All right, so this is our last lab, guys, in the whole class, lab 12. Thanks for hanging in to almost the end of the class. And this time we're going to study ecological communities. So you've seen this slide probably a dozen times by now, where we've defined communities as groups of populations of multiple species that are in the same place and the same time. I think I've shown you this picture. This is a restored prairie grassland near Chicago, Illinois. It's one of my favorite pictures ever. You can just see how beautiful and diverse communities can be. And so just to take a few more slides from the lectures, right? Communities are incredibly complicated. And I want you to get a sense for that complexity today. So if each species is a variable in a system, right? So, if you do a straight line, trend line in Excel, you get Y and you get X, right? There's two variables in that. So imagine a community with more than 1,000 dimensions. And to give you a taste for that, this formula gives you a way of calculating the number of possible pairs of interactions, right? So if you have S species, where there are P possible pairs that we can calculate in this way with this formula. Right, so you just stick in the number of species here and here, and then it tells you the number of possible pairs. And so because I do research out at the Ross Reserve, I know there's about 50 species of trees, which gives us 1,225 possible pairs of interactions. Right, so that would be like species 1 versus species 2, species 1 versus species 3, species 1 versus species 4, all the way to all 50 species interacting with each other. So you could imagine how difficult this would be to understand all those pairwise interactions, right, when you need 1,225 treatments without even any replication. Right, if we try to do 10 or 20 replicates, this quickly explodes to a number that's impossible to study. But the trees aren't the only thing out there, right? We were actually going to go out this week and study the understory spring ephemeral community. There's actually a ton of beautiful flowers out there. I'm really sad that I can't take you out there to see all these beautiful flowers, that they all flower before the trees close their canopies so they can do photosynthesis without any shade. But if we pretend that there's 100 of those, plus our 50 species of trees, then P goes up to 11,000 and change. But plants aren't the only thing out there, right? Plants, as we've learned in class, are interacting with vertebrates and invertebrates that might be eating them or potentially pollinating them or dispersing their seeds, so harming them or helping them. So if we add, say, 20 vertebrates, 100 invertebrates, I'm just making these numbers up, then with all of the plants we've already calculate, and you should probably check these numbers to understand where I'm getting them from. That would probably really help you understand. But we get 36,000 possible pairs of interactions. Microbes are incredibly important, right? Mycorrhizal fungi, nitrogen-fixing bacteria. If we say that there's maybe 200 of those, then we get 110,000 possible pairs of interactions. And all of these numbers I just sort of guessed at, right? They're probably all higher except this one, which I actually know. Right, so how do you begin to describe a system with hundreds of thousands of interactions and thousands of dimensions, right? We're way past rocket science here. And so what we're going to do today is we're going to think about species richness and a couple of ways that ecologists have tried to think about species richness and what it means. So I gave you this in lecture, but I'll give it again. It's just the number of species in an area. It's the simplest possible way we could describe a community. Just how many species are there? Right, so the idea is that you put down a quadrat. Remember in the lab last week, we put down a 10 meter by 10 meter quadrat and we counted how many individual trees there were. This time we're going to do smaller quadrats that are the size of a hula hoop. Like literally we usually use hula hoops. You lay that down and you can say, okay, well, if I look at this picture, I see purple coneflower. I see this species that I don't know the name of. There looks to be something here and maybe 1 or 2 species of grasses, right? So we found 5 species, let's say. If we go over here, maybe we find a different set of species, right? Some trees, maybe some riparian grasses. And so alpha richness is literally just the number of species that you find at some local scale. So I don't know how easy this is to see, but I put the Greek letter alpha into these two quadrats. So that's alpha richness. Ecologists also talk about gamma richness, right? So you can imagine that if there are different species here and here, then the total number of species you observed is actually greater than the alpha richness. And ecologists call this gamma richness. So it's the number of species at the regional scale. So we can put gamma in the whole field. And these are two ways of describing this community, right? How many species do we find at some local small scale, and how many species do we find at the landscape scale? And then ecologists also define something they've called beta richness, which is the weirdest of all of them, but it's sort of the rate at which you go from alpha to gamma. And so you literally just calculate it. You have to find what is the number I would multiply each alpha by to get to gamma. And so more intuitively, we can just rearrange this formula, divide both sides by gamma, and divide both sides by alpha, and we can get that beta is simply gamma divided by alpha. And so it tells you a little bit about species turnover, right? What is the rate at which the species composition changes as I go from here to here? But the problem then becomes, what area should we sample? We talked about this in lecture, right? If I double the size of this square, I'm going to find a different number for alpha, even while gamma stays constant, which is then going to change beta. So what do we do about this? Right, and I think I told you this in lecture, species richness, alpha richness and gamma richness, they both increase with the area that we sample, right? If I sample just this small area or the entire field, I'm going to get an incredibly different answer, right? Last week we used 10 by 10 meter quadrats. This week we're going to use hula hoop sized quadrats, right? And so if we sample a bunch of different areas, we can draw what's called a species area curve. And what ecologists have shown is that this increases logarithmically. As you sample one hula hoop, two hula hoops, 3, 4, 5, 6, right? You're basically doubling the area each time you sample a hula hoop. The cumulative number of species you've seen starts to drop off. And hopefully this is intuitive, right? The first place you look, everything you find is new. In the second place you look, you probably are mostly finding new species, but at some point you're approaching the gamma richness and you're not finding any more new species. And so this increases logarithmically. Sometimes ecologists talk about this saturating, but it doesn't actually, right? So if we take the log of both sides, right? So log S, if we take the log of this side, these are tricks that you probably don't remember from math, but what happens is if you take the log of this, the exponent comes down in front, and things that are multiplied together are added together in log space. And so I know this looks a bit weird, but what you've actually got here is now just a straight line where the x is log A and the y is log S. The slope is z and the intercept is log C. So it just becomes a straight line. And this is much easier to work with. You guys know how to do straight lines in Excel. You don't know how to do these kinds of equations in Excel. So this turns out, this linearized form turns out to be really useful. And this is what we're going to work with in lab today. But the problem is, this doesn't help us figure out what area to stop, because if we keep sampling bigger and bigger areas, we're just going to go to infinity. And so this is a problem that doesn't have a solution, in my opinion. And for the most part, I feel like community ecologists just ignore it. Yeah, so this is not an easy problem to solve. What area should you sample? Because as you increase area, you just get more and more and more species. And this means that the answer you're going to get is really going to depend on the size of the quadrat that you look at. So today we're going to explore the consequences of this by just making species area curves. We're going to go to a Purdue property where we would have gone last week called the Ross Reserve. If you know this golf course or you know the Ross Hills County Park, it's right adjacent to both of them. It actually has a laboratory out there in a log cabin. It's really cute. And what we normally do is we study a lowland area. So all of these topographic lines are basically showing you've got these flat uplands, and it all goes down and drains into the Wabash River. So there's a lot of creeks, like there's a creek running through here, and there's a creek running through here, there's a creek running through here. And so we're going to go down to one of those wet lowlands, and we're going to get a sense of how all that moisture potentially changes the communities. And then we'll go up to one of the upland sites. I think usually we go the other way around. We start in the upland, and we go down this path, and then we walk back to the bus. And up on the uplands, it's a very different community than it is down on the lowlands, and we're going to try to understand why that might be the case. And so the method we're going to use is called the Transect quadrat method. What we would do is, you learned about quadrats last week. So we're going to lay quadrats and count the number of species inside, but we're going to run a transect that's 10 meters long and then sample quadrats on at an interval along the transect. And what this does is, if you randomly lay transects using some mapping software or other tools, and then take quadrats at defined intervals, you get a nice sample of different parts of the forest. This is a method that ecologists frequently use to study communities and take field samples. And so what this allows us to do is draw our species area curve. We're going to do the linear form. I don't know why I put it this way. But just to give you a sense of what your data would look like, you would have done 5 quadrats in the upland, 5 quadrats in the lowland. What I ask the students to do, which you won't get a chance to do, is you can call the species whatever you want, but you have to use the same names, right? So if you say, this thing has a big stem, that's fine, call it big stem. But if you see it in the future, you have to keep calling it big stem, either both in the upland and the lowland, because they do share some species in common. And then what you would do is you just score the presence and absence of species. That's all you need. So just if there's 1 present, you can put a check mark or a 1, whatever you want. And so what we find then is that in the first quadrat, the cumulative richness is 2, right? So at that first area, we saw 2 species. Then we go to the second quadrat, right? Our cumulative richness now becomes 3 because we've seen this species before, but we haven't seen this species before. So we have 1, 2, 3 species that we've seen. Our cumulative richness goes to 3. So we go up. We go to quadrat 3, and this is all just made up data, right? We've seen this species already. So this doesn't add anything to our cumulative richness, but now we see 2 more. And so our cumulative richness goes to 5. Hopefully that makes sense, how the cumulative richness doesn't include things you've seen before, right? So if you don't understand how I got 2, 3, and 5 from this, take a look at your lab manual. There is some instructions on how that works. The cumulative area that we've sampled would have been the area of a hula hoop, which happens to be this many square meters, right? So the first area we sampled was the area of 1 hula hoop. You double it, because now the cumulative area is 2 hula hoops worth of area. The cumulative area here is 3 hula hoops worth of area, and so on. So that's where these numbers come from in the data that we'll look at in a subsequent video. So we take the cumulative area and the cumulative richness, and we should be able to make this graph. That's what you would have done with the class data. Now, just to get you the sense of alpha richness from each of these, alpha richness is the total number of species. So now the cumulativeness doesn't matter. We saw 2 species here, so we get 2. We saw 2 species here, so we get 2. Here we saw 3 species, so we get 3, right? What's the gamma richness from here? Remember, gamma richness is the total number of species that we saw. Well, that's going to be 5, right? Because the cumulative number is the total number of species we saw. We saw this one, 1. We saw this one, 2. We saw this one, 3. Didn't see this one. And then we saw these last 2, 4, 5. So the total number of species we saw was 5. That's our gamma richness. And then finally, we can calculate the beta richness, if this was our data, by simply dividing gamma by alpha, right? So alpha is 2, 2, and 3. It's just 5 over 2, 5 over 2, 5 over 3. Using this formula for beta richness, we can see that it's 2.5, 2.5, and 1.67. And really what this is meant to represent is the turnover of species, right? So you're getting about a 50% turnover in species as you go from here to here, and here we're getting some significantly more turnover in species. That's what you should be seeing here, right? Because we've got this turnover where this guy disappeared and this guy appeared as we went to the second quadrat. When we went to this one, these 2 disappeared and 3 more appeared. And so that's supposed to be what beta richness represents. Okay, so that is the pre-lab lecture. The next video is going to include just a demo of what we would have done in the forest to help you get a sense of what the activity would have looked like.